Morning, everyone. Today we are going to deal the revision topic, chapter 6, DNA. And DNA is acting as the genetic material. As you all know, the chromosomes. DNA is located in the chromosomes and they consist of genes which regulate the characteristics of an organism. So here we are going to discuss the descriptive characteristic features of DNA. So DNA is the genetic material in most of the organisms, and it carries the genes and it causes the inheritance of characters from one organism to another. But not in all the organisms, there are certain exceptions, especially in retrovirus virus. The genetic material is RNA. And in that case also, the genes are included in the RNA. So whether it is DNA or RNA, both it is made up of polynucleotide strands. And each polynucleotide strand again consists of the units that is called as nucleotide, which in turn made up of base pairs. So if it is DNA means there will be base pairs will be there because it consists of two polynucleotide strands. So apart from RNA acting as the genetic material, they are also having many other functions. That is, the RNA is acting as the structural component. How it is acting as the structural component means it is acting as the important component in ribosome where the protein synthesis is taking place. And along with that, RNA is also acting as the messenger RNA. That is, it will carry the information that is encoded within the DNA into an RNA that is called as messenger RNA, which will give directions for the amino acid assembly at the time of protein synthesis. So it can carry the message from DNA and it can be transported to the cytoplasm where the protein synthesis can take place. Then third function, when you are saying the RNA can act as adapter molecule. So how it is acting as adapter molecule means the RNA, a special type of RNA that is called as transfer RNA. It helps in transferring the amino acids from the cytoplasm to the ribosomes as per the codonic sequences that is given in mRNA. Then the last function, the RNA is also acting as the catalytic function. That is, in the ribosome, the 23S rRNA can act as the catalytic function. It can help in the protein synthesis. So all these four functions, or five. The RNA itself is genetic material. It can act as the messenger RNA. It can act as adapter RNA, and it can act as structural component. And at the same time, it is having catalytic activity, which is also called as ribozyme. So the both DNA as well as the RNA can act as the genetic material. So it is mainly the genetic material is made up of many nucleotides. The nucleotide itself consists of, the DNA consists of, if you are taking a human DNA, the number of nucleotides within that, it is 3.3 into 10 to the power of 9 base pairs in a haploid cell. If it is in diploid means it will be 6.6 into 10 to the power of 9 base pairs. And if we are taking a theta into 174 bacteriophage means the number of nucleotides will be 5,386, and in lambda bacteriophages it is 48,502 base pairs. And in bacteriophage, the first one, that is theta into 174, it is only nucleotide. There is no base pairs. We haven't mentioned because in that bacteria, the DNA, the genetic material DNA is single stranded. Whereas in lambda bacteriophages, it is base pairs. That means two polynucleotide strands will be there. Even in E. coli, which is the bacteria, that is in that the number of base pairs will be 4.6 into 10 to the power of 6 base pairs. So this is just a rough description, that is, in each and every organism, the number of base pairs will vary from one organism to another organism. So, the DNA, when they have found out within the nucleus, they were not clear about, the scientists were not clear about what it is made up of and how it is organized, how it is packaged within the cell, etc. So the pioneer step was taken to study more about the structural features of DNA. It was the study of Watson and Crick. It was came after that. It was came as the spiral helical model or spiral staircase model for the DNA. So what they said, each DNA molecule, it is located within the nucleus, within the chromosome, and each DNA is made up of two polynucleotide chains. And we have learned in last year that is a polynucleotide chain. It is made up of many nucleotides, and each nucleotide is connected with the help of phosphodiester, that is 5,3 phosphodiester bond. And each nucleotide in turn is made up of a pentose sugar, that is, in DNA means it is deoxyribose, RNA means it is ribose. The difference between deoxyribose and ribose is, in the deoxyribose, the second carbon, it is H only, oxygen is absent. Whereas in RNA, the second position, OH group, which is more reactive. And that is the main difference between deoxyribose and ribose. So this deoxyribose binds at the first carbon with the help of, with the nitrogen base. It can be either purine or pyrimidine. Purine itself again A and G, and pyrimidine it is C and T, cytosine and thymine. And in RNA means instead of thymine it is uracil. So this first carbon of the pentose sugar, if it combines, or it can combine with any of the nitrogenous base with N-glycosidic bond, and that forms the nucleoside. That means pentose sugar plus base, it is called as nucleoside. Then the nucleoside with phosphate group at the 5 carbon end forms means nucleotide. The neighboring nucleotides with phosphodiester, 3 carbon, will combine with 3 OH group of the third carbon will combine with 5 carbon with the help of phosphodiester. Then they can form the nucleotide, dinucleotide. Then again third one, if it is joining, trinucleotide. Like then many nucleotides with the help of phosphodiester, they can form polynucleotide strand. In RNA there is only one strand, whereas in DNA there is 2 polynucleotide strands. So Watson and Crick, they said each polynucleotide strand consists of it is made up of 2 polynucleotide chains, which is helically wound in a right-handed manner. And in that staircase, they compared it with a spiral staircase. In that staircase, the backbone is made up of sugar phosphate backbone, and the steps of that staircase is made up of nitrogenous base pairs between these 2 polynucleotides. And in that, the bases project inside. And these 2 strands in the DNA, they run in antiparallel. That means if one strand is 5-3 direction, means opposite strand will be 3-5 direction. Then the third one, they said, when the base pairs are formed between the polynucleotide strands, always this base pairs is between purine and pyrimidine. And there also specificity is there. The adenine always binds with thymine with the help of 2 hydrogen bonds. The same way, cytosine binds with guanine with 3 hydrogen bonds. Then the next point, what they said, the 2 chains are coiled in a right-handed manner. And since the base pairing bonding is between purine and pyrimidine, purine is 2 cycle, and pyrimidine it is only single cycle. So always the diameter of the DNA molecule will remain constant, that is 2 nm. Then the pitch of the helix, That means one complete turn, the length can be 3.4 nm. And one complete turn consists of 10 base pairs. So that means the distance between the neighboring base pair is 0.34 nm. Then because of these two strands which are anti-parallel and the base pairs also always complementary to each other, that particular structure, that staircase structure itself will give additional stability. That is mainly two factors only controlling or giving this DNA molecule more stability. That is, the first one, the plane of one base pair stacks over the other in double helix. So always the base pair, one base pair here means other base pair just one over the other. In that way, that gives them stability. Apart from that, these base pairs itself are connected by hydrogen bonds. So these hydrogen bonds as well as the arrangement of the base pairs one over the other gives them more stability. Then when we are, so this is proposed by this staircase structure, and that characteristic features were delivered by Watson and Crick. Apart from that, it was helped by Wilkins, Rosalind Wilkins. Her X-ray crystallographic studies gave them more support to come into the conclusion and elucidate the whole structure of DNA. Then when we will say the characteristic feature, here we have given the structure on the right side, the helical model it is given. So whenever the question they ask, you have to draw the helical to polynucleotide strands, and the coiling also you have to see, it is right-handed manner the coiling, and one full turn, that means 10 nitrogenous base steps also you have to draw, and each base pair the distance also you have to mark it, and the diameter also 2 nm you have to mark it. So polynucleotide strand you have to mark it, then sugar phosphate backbone you have to mark it, and then nitrogenous base pairs you have to mark it, and A and T means 2 hydrogen bonds, C and G means 3 hydrogen bonds. And if it is the detailed structure of DNA, the molecular level of DNA means the centermost figure you have to draw. That is sugar, pentagon shape, sugar. Inside that you have to write S. And the first carbon to the base. And that bond you have to label it as N glycosidic bond. Then the fifth carbon to the third carbon by a phosphodiester bond. And both the strands, one end of the strand should be 5 and the other end should be 3. Then the opposite strand, that the other end, it should be 3, and this end it should be 5. And all the labeling must be completed. So when the question is asked, name the bonds that is present in DNA. So DNA consists of N-glycosidic bond, phosphodiester bond, and hydrogen bond. Now we'll move on to the next concept, that is Irwin-Chargaff's base pairing rule. Along with this, the scientist Irwin-Chargaff, he proposed the rule. That is, he said the ratio between adenine and thymine and guanine and cytosine are constant and always equal to 1. So from this itself, it is understood that the total amount of adenine will be equal to thymine, as well as the total amount of cytosine will be equal to guanine. And the next one, it is the ratio between adenine and thymine and cytosine and guanine is constant, or we can say specific for each species. So human beings means there will be a specific ratio, and that will be varying from other animals. That is, lion means another, plants means another. So each and every species will have a particular ratio or a specific ratio between the A and T as well as C and G. And that ratio will vary from one species to another. So all these concepts together, they comprised as the Irwin-Chargaff's base pairing rule. So the total amount of purine will be equal to total amount of pyrimidine. Then A plus G by T plus C is equal to 1. Then A plus T divided by C plus G, that ratio will vary from one organism to one species to another species. One species to another species. The next one it is we have learned that in all the genetic material, all organisms, majority of the organisms, DNA is acting as the genetic material. Then how this DNA is controlling the characteristics of an organism? So in all organisms, the characteristics will appear either in the form of a particular trait, or that trait in turn is controlled by the protein. So it can be either in the form of a protein, or sometimes some metabolic activities which is mediated with the help of an enzyme. So either the protein production or the enzyme production. Enzymes, all the enzymes are protein. So to summarize, the protein production or the synthesis of the protein is regulated by DNA. How it is regulated, or the genes which are present in the DNA. So here the Crick, Francis Crick, he proposed a theory that is called as central dogma. So what is central dogma means? It is the unidirectional flow of information. It is the unidirectional flow of information from DNA to RNA, then ultimately to protein. So he said the protein synthesis that is taking place within the cell, it's all controlled by DNA, whereas the protein synthesis is in cytoplasm and the DNA is located in the nucleus. So how this is possible? So DNA is making a small copy of a portion of the DNA, and that is called as mRNA, messenger RNA. So the genic sequences or the nitrogenous base sequences is copied into another molecule that is called as messenger RNA. This messenger RNA from the nucleus, it will transport into the cytoplasm. And as per the instructions or as per the base sequences in the mRNA, the amino acid assembly will take place that leads to the formation of protein synthesis, which can be expressed either in the form of enzyme or in the form of a particular type of trait. Sickle cell anemia, sickle trait, RBC and all. So all these are controlled by DNA. So if any defect in the DNA or any deletion or addition in the DNA, that can alter the structure of RNA, which can alter the characteristic feature of protein. And their nature can be altered. So that will be reflected in the form of many disorders. So here he said it is the unidirectional flow of information from DNA to RNA, then to protein. When we discuss this one, there is another category, that is virus, where we can call it as retrovirus. In retrovirus, instead of DNA, there is no DNA at all because their genetic material is RNA. So in such cases, how the protein is regulated, synthesis of protein or viral proteins are regulated. So that is framed by another scientist. His name is Temin. It is also called as Teminism. So how, even though there is no DNA in virus, and virus always require a host cell to multiply. So how they are multiplying, how they are making their own proteins in the host cell. So whenever the virus attacking the host cell, the RNA, the genetic material of the virus, that is RNA, will enter into the host cell. There, it will take initiative for the synthesis of DNA. That means here a reverse process. In all other organisms, DNA to RNA. But here in virus, it is in a reverse direction. That is, RNA takes initiative for the synthesis of DNA with the help of an enzyme that is called as reverse transcriptase. That process is called as reverse transcription. So as a result of reverse transcription, RNA makes a DNA. Then DNA takes initiative through the mRNA and protein within the host cell. So within the host cell, the viral proteins can be synthesized. So that process is called as reverse transcription. It is proposed by Temin. So these flowcharts are very important. Even central dogma, when the question is asked, means you have to write that flowchart. That is the previous slide I have shown this flow chart, and you have to explain. It is a unidirectional flow of information from DNA to RNA. So DNA to RNA, the process is called as transcription. And mRNA to protein, the process is called as translation. And DNA to DNA, that is replication. Okay, so that flow chart is very important. Then DNA, RNA, protein, that concept, unidirectional flow of information. That entire thing we will call it as central dogma. And central dogma reverse, already I said, that is in virus. Reverse transcription, that process is called as, that concept is proposed by Temin, hence that process is called as Teminism. Then we will move on to the next one. So already we know that DNA is located within the nucleus. So each and every cell in our body, it is very, very microscopic. So through the microscope, we cannot see the nucleus and inside the chromosomes, etc. Ordinary microscope, we will not be able to see. We need more powerful electron microscope. So when the scientists, they observed through the electron microscope, they have found out that is the DNA. Later they came to know the length of the DNA. In a human cell, the DNA can occupy approximately around 2.2 meters. More than 2 meters length, a very thin thread. The nucleus itself through the microscope we won't be able to see. So within that nucleus, how this 2.2 meters of DNA is closely wrapped inside. So that process is called as packaging of DNA. And the packaging of DNA in prokaryote is different as well as in eukaryote it is different. The prokaryotic cell, when we are studying, as we all know, it is a special type of cell where there is no definite nucleus. There is no nuclear membrane, nucleoplasm, etc. Instead of that, the DNA of prokaryote itself is special because it consists of only a single DNA. And it is double stranded as usual, but it is not wrapped by proteins. It is naked circular DNA. It is only one DNA that is naked. Naked means there is no wrapping with histone proteins. The protein wrapping is absent. And that too circular. There is no open ends. And it is just scattered within the center region. That region is called as nucleoid. We won't call it as nucleus. There is only nucleoid region. And it is scattered or organized in the form of a large loops held by proteins. That is not histone proteins, held by proteins. A large loop in that form. So there is not much packaging is essential for the prokaryotes because that only single cell. There is no compartmentalization between cytoplasm and nucleus. There is no definite nucleus. Only there is single DNA, double stranded naked DNA is held in the form of a large loop. Whereas eukaryotic cell, when you are saying, there is a distinct compartmentalization, nucleus and the cytoplasm. The nucleus encloses nucleoplasm. Within the nucleoplasm, the DNA is arranged in the form of small thread-like structures, like a woolen thread. Like that, it is scattered in the form of network of fibers that is called as chromatin fibers. And this chromatin fibers, how they are arranged, that is together, we'll call it as packaging. So, when we go deep into the microscopic structure, we can say the DNA is wrapped with the help of a special type of protein. That protein is called as histone. And what is histone protein means? It is a basic protein and it is positively charged, whereas DNA is negatively charged. So there will be a force of attraction between positive and the negative molecules. And this histone protein itself consists of or arranged in the form of held together in the form of histone dimer units. Actually, 8 subunits are there. Since there is 8 subunits, that is all coming under the category of histone, it is called as histone octamer. In that histone octamer, 4 subunits forms an upper tier and other 4 forms a lower tier in the form of dimeric configuration. So 4 balls, the figure it is given on the top. So 4 subunits together, one tier, one row, and another 4 lower row. And they are named in different way, that is H2A, H2B, H3, H4. So H2A, H3, H4, H2B. So that 4 together forms one component, one tier, and like that, same like this, another one also. So altogether 8 subunits together in the arrangement, arranged in the form of a ball-like structure, and that is called as histone octomer. And on that octomer only, 2 coils of DNA, approximately 2 coils, even though one 3/4 coil only is there, but approximately 2 coils of DNA. That is in an which will say the coiling is somewhat like anti-clockwise coiling. Anti-clockwise coiling. So one strand will enter, and after 2 coiling, the other end it will go out. So 2 coils of DNA will be there. These 2 coils of DNA around this histone to the histone octomer is connected by another type of histone that is called as H1. And the DNA will go out. That will not have a particular region of the DNA will not have the histone proteins. Then after a particular distance, then again they will coil around the next histone molecule. So the DNA that is present between the neighboring histone molecules, that region it is called as linker DNA. So the histone octomer with 2 DNA coils along with H1 and the linker DNA together, we can call it as nucleosome. So each nucleosome consists of approximately 200 base pairs. In that, 174 bases for 1/3/4 of the coil, and the remaining forms the linker DNA. So each nucleosome, when we say the number of nucleotides, approximately we can calculate it as 200 base pairs. So many problems related with that you can expect for the board exam. So in a DNA, this many nucleosomes are there. So calculate the number of base pairs. Or in the reverse way, in a human genome, 6.6 into 10 to the power of 9 base pairs are there. So calculate the number of nucleosomes. Like that way different questions can be asked. So the packaging, it is the only the primary packaging. So like that the nucleosomes will be formed. This nucleosome units again further coils. Further coils, so here I have given. So the nucleosome with the linker DNA, neighboring nucleosomes in between the linker DNA, that gives the characteristic feature that is called as the bead-on string structure. In that way, they will be arranged in the form of, again, they further coils. The bead-on string structure further coils, and then they form the structure that is called as chromatin fibers in the form of a thread-like structure that will be present within the nucleus. So in a typical interphase cell, always the DNA along with the histone octomer, that is in nucleosome organization, along with that they will be arranged in the form, and that is called as chromatin material. But when the cell is about to enter into the division, that is means the end of towards the end of prophase, there the condensation of chromatin fibers will take place. So at that time there will be one more level of coiling, that is called as that is with the help of non-histone proteins. So the chromatin fibers will again held tightly or coiled tightly with the help of non-histone proteins. And then they will acquire the characteristic rod-shaped structure. That time we'll call it as chromosomes. So we have learned prophase, the chromatin fibers, and towards the end, the prophase, the coiling condensation starts. And by the when they enter into the metaphase, each chromosomes will appear maximum condensed, maximum rod-like structure. So that is with the help of non-histone proteins. And when we very closely observe after staining, we can see certain regions of the DNA, they are lightly stained and certain regions are darkly stained. So based on that staining, it is named with two names, that is heterochromatin. Hetero means differential colors, chromatin, chromo, color. And why certain regions are tightly bound and why some regions are lightly stained. So the darkly stained, densely packed region is called as heterochromatin because there the coiling is very firm and that particular region is inactive. It is not taking part in the transcription. Whereas in lightly stained region, that is called as euchromatin. In that region, we can see it is loosely packed and lightly stained. This loosely packed and lightly stained regions, we can infer that that particular region, the DNA is unwinding. Unwinding of the DNA. That means histone proteins and non-histone proteins and all will be removed and the DNA is about to separate and one of the strand will act as the template and from there the RNA will be forming. That process is called as transcription. So wherever the light stain areas are there, that areas are called as euchromatin. That means that particular region is taking initiative for the synthesis of RNA transcription. Now we'll move on to the next topic that will be discussed in the next module. So with this, I am concluding. Thank you very much.